Hello, my name is Celia. I'm a researcher at LMMP PUC-Rio, and today I'm going to present the current work developed with Rafael Ribeiro, Bruno Liupercio, and Márcio Carvalho. This work consists in the study of PDMS microcapsules for triggered release by osmotic pressure. Microcapsules can be used as a vehicle for triggered release in many different applications. Microcapsules are formed by a core material involved in a shell. Depending on the application, microcapsules are produced with specific properties considering the trigger mechanism used, so that their content is released at desired time or location. The triggering mechanism can be osmotic pressure, temperature, pH, or external stress. In our study case, we will focus on the osmotic pressure trigger. The release of the active compound that is located in the core of the microcapsule occurs when the trigger causes a mechanical rupture of the cross-link cleavage or the polymerization of the shell. The fact that we can use microcapsules in different industries such as pharmaceutical, cosmetics, foods and beverages, and also in enhanced oil recovery in a controlled environment brings our research to future technology innovation. So, the goals of this work are to produce PDMS microcapsules with different amounts of salt in the interface, vary salinity differences between the microcapsules and the medium, which will consist in two different studies, Hypertonic and Hypotonic, to observe the microcapsules volume variations triggered by the osmotic pressure, and finally acquire and analyze 2D and 3D images using the Confocal Microscopy technique. So, let's start with microcapsules production. The water in oil in water double emulsion microcapsules were prepared using a co-flow and flow focusing capillary device. The first diagram illustrates the dynamic of the microcapsules production, where the inner phase goes inside the injection capillary, the middle phase and the outer phase flows through opposite sides, so the middle phase flows outside the injection capillary and the outer phase flows outside the collect capillary. Microcapsules with different diameters and shell thicknesses are formed, controlling the flow rate of each phase, and flow in the collect capillary to the collect phase. For better understanding, on the video recorded with a high-velocity camera, it is possible to observe the production of the microcapsules produced in this work. The composition of the inner phase and the collect phase change according to the experiment, if it is hypo or hypertonic medium. The osmolality at the inner phase and the collect phase should be the same to set an isotonic medium. The microcapsule shell is formed by PDMS polymer in a 10:1 ratio. The outer phase is 10% PVA solution that acts on the shell formation. A confocal laser scanning microscope was used for visualization of the microcapsules during the experiments. The advantage of using this type of microscope is that it allows obtaining 3D images of microcapsules for volume quantification. Also, a concavity slice was used to enable multiple tests simultaneously. To use it on the microscope, a 3D model was designed and printed in lab facilities. This device improved the quality of image acquisition. Confocal microscopies are commonly used in the biological area in order to analyze 3D samples. In order to produce the 3D visualization, the equipment relies on a pinhole filters which restrict all the light from out of focus planes and let only the focal plane light to pass through, as shown by this image. So, each section of the sample is acquired separately and later reconstructed to form a 3D image. This 3D image on the right-hand side of the slide is an example of an image composed of a stack approximately 60 plane images. Using this technique, it is important to add fluorescent pigments inside the microcapsules. This will allow to distinguish the inner phase from the capsules. Now, let's see the results of the hypertonic medium tests. To perform the hypertonic tests, microcapsules with low osmolality were prepared. These microcapsules have 55 milliosmol per kilogram of osmolality. The test was conducted using 5 cavities of the device. Each cavity was filled with water and salt. with different osmolality values. The lower value was 55 milliosmol per kilogram, and the highest value was approximately 4,000 milliosmol per kilogram. It was expected to observe microcapsules buckling with the increase of the osmolality. This image shows a single focal plane from the results obtained during the hypertonic tests for each osmolality. The acquisition was performed along 10 hours within intervals of 3 minutes. These results show that microcapsules shrink and the shell buckles. The inner phase content is represented in yellow, and the middle phase, the shell, is represented in pink. The image was acquired in the highest fluorescent intensity plane, which means the middle plane at the initial time. As qualitative evaluation, we conclude that in the cavity of control was observed deformation, justified by the isotonic medium, and also after 10 hours, we can see that the minimum amount of the content inside the microcapsule was reached for the higher osmolality difference medium. The most interesting behavior, it was a loss of the fluorescence intensity with increase of the osmolality at the inner phase. This means that we successfully saw the content release. At 10 hours of experiment, we just observed the microcapsule shell in the higher osmolality difference medium. For quantification, 3D images were reconstructed from images of different focal planes. The results were obtained by treating different planes from 3D images that allows analyzing the capsule's volume. In the graphic, we see that microcapsules exposed to a higher osmolality have greater volume variations. The blue and orange curves show a volume reduction of approximately 55%, while the other two cases indicate the size reduction of approximately 90%. The results showed an almost total loss of content for microcapsules immersed in high concentrations of salt, which indicates a promising tendency for larger samples. And now I'm going to present the results of the hypotonic medium tests. To perform the hypotonic tests, microcapsules with a high osmolality were prepared. These microcapsules have 2,000 milliosmol per kilogram approximately of osmolality. The test was conducted using 5 cavities of the device. In each cavity was set a different osmolality value from the highest value to the lower value. It was expected to observe microcapsule swelling and the rupture decreasing the osmolality. Microcapsules that underwent hypotonic tests show similar behaviors for different osmolalities. There is a constant. Swell, which is more pronounced when the osmolality of the medium is smaller. This image showed the microcapsule exposed to a medium of 200 milliosmol per kilogram, which is the minimum concentration used. Although a large swell was observed, even after 7 days of exposure, microcapsules bursting was not observed. Note that the scale of the images are different. On the right-hand image, microcapsules were bigger than the ones in the left image. Also, the capsule in the middle has approximately the initial volume when all the others grow. Proportionally, microcapsules have grown more than 2 times the initial diameter. The microcapsules don't grow with uniformity. They are not perfectly concentric. They grow from the thinnest shell part, which is more permeable. These results were obtained in different planes from 3D images. This allows us to do an analysis of volume. Through the graphic, we see a smooth swelling of microcapsules when exposed to mediums with high osmolality values, yellow curve and gray curve, with a volume increase of 29% and 41% respectively. The increase is even higher, 52 and 72% respectively, for the mediums with lower osmolality, orange curve and blue curve. So, the medium with lower salinity values are associated to the higher increase of microcapsule volume. Moreover, we can see that the microcapsules in lowest osmolality medium grow faster than the other cavities. Although a large swell was observed, microcapsules bursting was not observed. This suggests that the microcapsules with a PDMS shell is too elastic, and probably only microcapsules with an inner content with osmolality even higher than the used will burst. In summary, we were able to produce PDMS microcapsules with different amounts of salt at the inner phase. The effect of microcapsule properties using the osmotic pressure trigger was successfully studied by tracking their volume variations using confocal microscopy. The results indicate that it is possible to trigger the inner phase content release of the microcapsules, exposing them to a medium with solute concentration approximately 3.5 times higher than the inner phase solute concentration. And finally, results also shown the microcapsule swelling when exposed to a medium with lower solute concentration. The swelling mechanism observed is controlled by microcapsules properties such as diameter or shell thicknesses. I would like to thank Shell and ANP for funding this research. Thank you all for your attention.